Hello everyone. Are I'm here to explain the photosynthesis. We are in the part 5 of this lecture. In this lecture, I'll be dealing with the dark reaction. So let's start. First of all, in dark reaction, it happens in the absence of light. It is not at all like that. It is just the myth and nothing. Dark reaction also is somewhat affected by the presence of light, because dark reaction depends on the products of the light reaction. So if light reaction stops, after some time the products of the light will also finish, then automatically dark reaction also comes to an end. So dark reaction is just a name that, and it has nothing to do with absence of light. Okay, it is also affected by the presence of light also matters to it. Now dark reaction is also known as the biosynthetic phase, and here basically the products, whatever products we read in the cyclic phosphorylation, non-cyclic phosphorylation, we got ATP and NADP as the products. So here these products are going to be utilized basically in the dark reaction. Okay, we can say that the products of the light reaction are used here as the reactants of the dark reaction. So that is all what they have mentioned in this section. And along with this, we can also say that light reaction, it has three products actually: ATP, NADPH and O2. Okay, and all these are mentioned over here. Now coming to Melvin Calvin, he used radioactive carbon 14 in algal photosynthesis, and he led to the discovery of the first CO2 fixation product, which was an organic acid of 3 carbon. And he also contributed to working out the complete biosynthetic pathway, hence it was called Calvin cycle after him. The first identified was 3-phosphoglyceric acid, or in short PGA. So scientists came to know that okay, there is a carbon product, 3 carbon product as PGA. So they did a wide experiment on the other plants as well to figure out if there is any other product. Then they find that everywhere the first stable product that they find after CO2 fixation is a organic acid compound. But they also came across another section where that organic acid turned out to be a 4 carbon atom instead of a 3 carbon atom, and that 4 carbon atom compound was named as oxalic acid, OAA, first stable product after CO2 fixation. So they divided the plants, that this plants follows two different pathways, C3 pathway, C4 pathway, and depending on that they distinguished the plants. The plants CO2 fixation, two different pathways, they distinguished the plants as C3 PGA is the first stable product after CO2 fixation, and in C4 as OAA, oxalic acid 4 carbon compound. Now they wanted to know the primary acceptor of CO2. Okay, that they knew that one CO2 will come, but who is the one accepting the CO2? Okay, the number of carbon atoms it possesses. So they thought, since in C3 the first stable product is PGA, is a 3 carbon compound, so obviously the primary acceptor must be a 2 carbon compound. Then one carbon from CO2 will come, and they both are going to be total of 3 carbon and form a product of 3 carbon compound. But it turns out to be completely different. When they do a lot of experiment, they come across that actually it isn't a 2 carbon compound which is accepting the CO2, it is a 5 carbon compound. It is a 5 carbon compound that is accepting one CO2, and that 5 carbon compound is RUBP. So regarding RUBP, ribulose bisphosphate, you have come across Rubisco in the enzyme chapter. So there Rubisco was known as the most abundant in case of whole biosphere. Okay, most abundant protein in case of whole biosphere, and in case of, and there was collagen also in case of plant world, as far as I remember. So please remember that whenever you're reading biology, wherever you get a chance to revise back, recollect back from something previous, you must do that. Then automatically you will be revising also, because this is a way of studying biology. Biology is not like maths that you learn the concept and okay. Biology is about how many times you revise, how many times you can recall, that many times you'll remember. So RUBP is a ketose sugar, 5-carbon ketose sugar, that is all mentioned here. And this was what they thought or they believed, but actually unexpectedly what came out. So this is all about this. You will read it on your own and you will understand that everything I have explained it already. Now coming to Calvin cycle. Depending on Calvin work only, they worked, Calvin and his co-workers worked out the whole pathway. So on that regard only, the name was given as Calvin cycle. Okay, so here RUBP will be regenerated, which we'll get to see in the cycle itself. So in the cycle we will discuss. Calvin pathway occurs in all photosynthetic plants. See, plants may either go through C3 pathway or it may go through C4 pathway. But all the photosynthetic plants, irrespective of which pathway they follow, they will surely undergo this Calvin pathway. Okay, both the plants will undergo Calvin pathway. So later on we'll understand the meaning of this statement more. When the right time will come, I'll let you know. Now coming to the 3 steps of the Calvin cycle. First is carboxylation. Second is reduction, and the third is regeneration. This is the cycle as you can see in the textbook. So first we will just go through this cycle so that the theory part we can understand well. So here this RUBP, ribulose 1,5-bisphosphate, is actually a 5-carbon compound. So now the CO2 fixation will take place, 1 carbon. So 5 carbon plus 1 carbon, 6 carbon compound we should have obtained as the first stable product, but instead we know that the first stable product is a 3-carbon compound, that is PGA, phosphoglyceraldehyde. Okay, sorry, 3-phosphoglyceric acid. 3-phosphoglyceric acid. So how come 6 carbon and 3 carbon it's not matching? So we say that we get 2 molecules of a 3-carbon compound. We get 2 molecules of PGA. That is why in that terms we say that we get the first stable product as PGA, 3-carbon compound. But 2 molecules of the 3-carbon compound we used to get. Okay. Then the next step will be reduction. And in this reduction step only, what will happen? See, this is 3-phosphoglycerate. This is the first stable product. Okay. You can say it's 3-phosphoglycerate or 3-phosphoglyceric acid, PGA, whatever, 3-carbon compound. Okay. Now, coming to reduction part. The second step. Here in the reduction, we'll get the glucose and whatever food we wanted, we'll get that. Here in this step is very important. Why? Because here you will see utilization of ATP and NADPH. The products of light reaction will be here utilized. Okay, as I already mentioned, in the dark reaction they have their role. This is the exact place where they will have their role, and here ATP and NADPH will be utilized, and this way the reduction of 3-PGA will take place. And after reduction there is going to be regeneration. But prior to that, let me tell you, for this reduction, in one turn, in the reduction step, we will need 2 ATP and 2 NADPH. Okay, 2 ATP, 2 NADPH. And after reduction, when you go to regeneration of RUBP, regeneration of this RUBP is needed. Why? Because if RUBP will keep on regenerating, then only this cycle will go on taking place. Okay, that is why regeneration of RUBP is needed. So in the regeneration, here 1 ATP will be again utilized and regeneration RUBP will take place. So if overall we see in this entire cycle, the most important part of the cycle is, first of all, we need to know only what will be the first stable product and how many carbon atoms it possesses. And what else we need to know is how many ATP and NADPH will be utilized in this entire cycle, in the second step, in the third step. Individually we need to know that. So if we see that in the whole cycle, total 3 ATP and 2 NADPH will be utilized in one turn of this Kelvin cycle. In the second step, we will need 2 ATP and 2 NADPH will be utilized, and in the third step, 1 ATP will be utilized. So overall, if we see 2 ATP plus 1 ATP, total 3 ATP, and here already I said 2 NADPH, so 2 NADPH. Okay, 2, 1, 3 ATP, 2 NADPH will be needed in one turn of this Kelvin cycle. If we ask individually in the second step, then you will say 2 ATP, 2 NADPH will be utilized, and if they ask about third step, then you will say 1 ATP will be utilized. This way also, not necessarily it always will ask you entire cycle, you tell me how much utilized. They may individually they used to ask also in the questions that tell me in the second step of Kelvin cycle how many ATP is utilized, or in the third step. So that way questions are possible. So just in the figure, the detail is given: carboxylation during which CO2 combined with RUBP. Ribulose 1,5-bisphosphate means RUBP. Reduction during which the carbohydrate is formed at the expense of the photochemically made ATP and NADPH. Regeneration during which the CO2 acceptor RUBP is formed again, so that the cycle keeps on continuing. So one read, let's make it quickly. Carboxylation, fixation of CO2 into a stable organic intermediate. Remember, carboxylation is the most crucial step of the Kelvin cycle, because CO2 will be utilized for carboxylation of RUBP. So now the catalyst of this reaction is also important to be highlighted from this part. The enzyme is RUBP carboxylase. Okay, as I said already, 2 molecules of 3 PGA, 3 carbon compound, 2 molecules of 3 carbon compound will be formed. As it was 6, no, so how will you balance it? So 2 into 3 carbon. Okay, since this enzyme also has an oxygenation activity, so it would be more correct to call it RUBP carboxylase oxygenase or Rubixco, because it has two properties. It has carboxylation property as well, oxygenation property also, but one property only it will represent at a time. Okay, but usually it is known as Rubixco only, but it will only show one nature at a time, either carboxylation or oxygenation. Okay, reduction, series of reactions that lead to the formation of glucose. The step involves utilization of 2 molecules of ATP for phosphorylation and 2 of NADPH for reduction per CO2 molecule fixation of 6 molecules of CO2, and 6 turns of the cycle are required for the formation of 1 molecule of glucose. So in this cycle, to get 1 molecule of glucose, you have to conduct 6 turns of the cycle. 6 CO2 has to be fixed. Okay, 6 CO2. How many CO2? 6 CO2 has to be fixed, and 6 turn of this cycle will have to take place in order to get 1 molecule of glucose, because 1 glucose is molecular formula is C6H12O6. So carbon 6, isn't it? So that is why 6 carbons will be required. So 6 molecules of CO2 will be needed for fixation, and as a result 6 turns of the cycle will take place to get 1 molecule of glucose from the pathway. Now comes to regeneration, the third step. Regeneration of CO2 acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. That is why regeneration is required. Here same thing only. Earlier whatever I said, overall 3 molecule of ATP and 2 molecule of NADPH will be required. That is it. This is a chart. This is made for 6 turn cycle, not for 1 turn. Here whatever is written 3 molecule ATP, 2 NADPH, this is for 1 turn. And this chart is written for 6 turn because 6 CO2, 1 glucose. So here this is how this chart came. If I explain you, then I'll show you this chart which I have made. See, this one is for 1 turn. 3 ATP will be utilized, 2 NADPH will be utilized. These are utilized and this we will get after that. We have nothing to do with this. We just need to know these numbers, okay? And then for 1 turn, and since we will need 1 glucose, so 6 turns will have to take place. Okay? That way 18 ATP and 12 NADPH. Sometimes they will ask you directly, for 1 glucose how many ATP is utilized. But if they ask you for 1 CO2 fixation how many is utilized, so question, depending on the question you have to answer. So they will try to play with the question in this type of chapters, okay? In this topic they can play with the question, how they will frame the question. They can trick you that way in the language part. Okay, coming to Calvin cycle, as I said, second step 2 ATP, 2 NADPH, third step 1 ATP. All total, if we see in the Calvin cycle, 1 turn, then 3 ATP and 2 NADP each. And here 1 glucose, 6 turn. This is something I already mentioned. And this one ahead of it I mentioned. So this is all for your Calvin cycle. Thank you. And one more thing, do not ignore this Calvin cycle because 5 mark question it might come. So you have to properly answer it. And while answering, remember to draw this also. It will also carry marks. Then stepwise you have to write down like your book only. But do draw. I'm pretty sure you will get. Even if not 5, 4.5 for sure you will get. Okay. And 0.5 will depend on your presentation, how you have presented it. I hope you have liked the video. Thank you.